My name is Higgs and this is my friend Boson. Welcome to the Higgs world.
Thanks for the introduction, Higgs. In this presentation, our goal is to offer you a quick summary of the research and conclusions. We follow inquiry-based learning model where students take lead in learning.
First, they get a quick review of the research via the videos. Second, they review the paper to learn and understand key concepts. Finally, they can follow up with further research on their own to dig deeper. In the end, students would have learned something new, latest and relevant. Isn't that wonderful, Boson?
Great point, Higgs. Please follow the links to review the research in further details. We want to thank all the authors and publishers for making this research public, thereby advancing science and innovation. Please like, share and subscribe.
Today, we are reviewing a paper titled, Interaction Between Disks Large and Pins, LGN, GPSM2. A Comparison Across Species. Disks large is a multifunctional scaffolding protein. Disks large has been identified as a factor involved in orientation of the meiotic spindle.
The orientation of the meiotic spindle determines the direction of cell division, and therefore contributes to tissue shape and cell fate. Interaction between the multifunctional scaffolding protein, disks large, and the canonical spindle orienting factor GPSM2, called pins in Drosophila and LGN in vertebrates, has been established in bilaterian models, but its function remains unclear.
That is so intriguing, Boson. Researchers used a phylogenetic approach to test whether the interaction is obligate in animals, and in particular whether pins, LGN, GPSM2, evolved in multicellular organisms as a disk's large binding protein.
Research shows that disks large diverged in C. elegans and the syncytial sponge Opsacus minuta, and propose that this divergence may correspond with differences in spindle orientation requirements between these organisms and the canonical pathways described in bilaterians. Remember, that meiotic spindle determines the direction of cell division, and therefore contributes to tissue shape and cell fate.
That is a very interesting observation, Boson. Researchers also demonstrate that pins, LGN, GPSM2 is present in basal animals, but the established disk's large interaction site cannot be found in either Pachyzoa or Porifera. Pachyzoa are a basal form of marine free-living multicellular organism. Sponges, the members of the phylum Porifera, are a basal animal clade as a sister of the diploblasts.
Results suggest that the interaction between pins, LGN, GPSM2 and disks large appeared in cnidaria, and therefore it may be speculated that DLG may have evolved to promote accurate division orientation in the nervous system. Cnidaria is a phylum, containing over 9,000 species found only in aquatic and mostly marine environments.
The findings presented in this study indicate that the mechanisms of spindle orientation have undergone substantial evolution within the animal kingdom. Interaction between pins, LGN, GPSM2 and disks large appears at the origin of neurons, suggesting it may have served as a template for asymmetrical cell division regulation in organisms, with increasingly diverse and complex tissue development. That was an interesting paper. Hope you liked it, Boson.